Yes, very well.
Okay. Yeah, thanks, Wei Ching. I feel like I have drawn the lucky straw here today. Hi, folks. Good afternoon. Lucky because I can't claim to have seen a lot of patients with cardiac amyloidosis, but talking to you about genes is something I'm quite familiar with. So very happy to talk to you about the genetics that I know around amyloidosis. So the talk is actually a lot broader. I felt that this could be a great opportunity to start at a very broad base to introduce to everyone again the concept of protein coding genes being the indivisible unit of inheritance, reminding everyone that there are about 20 to 25,000 of these protein coding genes in our genome. So of course, you recall from your medical school days, each gene is a string of bases. Bases are the chemicals that make up the DNA. ACGTs are the canonical bases. And each gene is therefore a string of these letters or bases. So if you look at the genome in the linear layout, all the genes are laid out in series, and they can be of varying lengths, as you can see here. And then there are also distances between genes, and these make up your intergenic region. So tens of thousands of bases can lie in between genes. And if you add up all of that together, the bases between genes and the bases inside genes, it literally is 3 billion bases altogether. So that's one single human genome, which is very vast. And if you take the analogy that's frequently mentioned in our space, it's analogous to 1,000 Bibles. And if you went to the Wellcome Trust Museum down in London, there is this one cupboard where a single human genome is put into all of these volumes. And as you can see here, there are 5 volumes of chromosome 11. And if you open any of these books, this is literally what you see, the 4 letters in the inscrutable code of the human genome. So having said that, each gene is important because it confers function to a cell. Many genes participate together, produce this function. There are some genes that are especially important because when they are mutated, they will cause damage to function. So this is exactly what one of the genes that we're talking about today, which Yok-Chin mentioned, TTR gene. And then there are other genes that when they are actually mutated, they will only cause mild functional change. So these are more easier to think of them as being modifier genes. So another good analogy that's frequently used in this context is the pipe organ. If you take out one single pipe, it completely screws up the organ. Or you could have a pipe that's just playing slightly out of tune. So in the whole vast space of where genes underpin diseases, I feel that this slide, I'm now using it so frequently, forgive me if you heard me use this slide before, it really helps to understand that diseases lie on the spectrum of common on one end and rare on the other. And in the space of common diseases, they are polygenic, they are multifactorial. It's not just genes, but also environment, diet, smoking, and so on. Whereas at the other end of the spectrum, which is rare diseases, and we're talking here about amyloidosis, which is a rare condition in the context of hereditary amyloidosis, then it's one gene, one disease, a monogenic condition, highly penetrant, highly inherited in our cardiovascular world, mostly autosomal dominant. So there is considerations going for gene therapy. And then the key, again, to remember is that there are common variants that impact upon common diseases on the other side, and rare singular variants that impact upon rare diseases on one side. And then, as you heard, in most cases, common and rare diseases can look very similar. Hypertrophy, for example, can look very similar. But the key is that in the rare spectrum, these are caused by single gene mutation. So again, this is a slide that I've shown before in previous talks. Forgive me if you've seen it. The easiest way that we think about it is breaking it down into muscle, big vessels, electricals, and then more recently, another few categories because the genes have been found for some of these. So hereditary amyloidosis obviously falls here. And again, there are very good guidelines now for genetic testing in our clinic, class 1 indications for HCM, and all these electrical conditions as you see here. And the one that Yok-Chin so nicely kicked us off today with hereditary transthyretin mutation. So I think the key for why genetic testing has become so accessible is the onset and the advent of next generation sequencing. And today, literally, if you follow the literature and maybe you purchase stocks on the stock exchange as well, Illumina is the world market leader with a sequencing machine that doesn't look that different from a microwave, for example, that can sit on the desktop counter. So in the realm of sequencing, you could really sequence the whole genome. That's your 3 billion bases of an individual. Or you can sequence the protein coding genes. These are the 20,000 that we spoke about. Or you can sequence the panel of genes, say a panel that deals with hypertrophic cardiomyopathy, or a single locus, if you know exactly which variant it is that this family has for its inherited condition. And the good thing is that the world has come together in the form of the American College of Medical Genetics guidelines as to how to assess variants in each gene to confer and classify them as pathogenic. So every variant that comes out looking different from a human reference would be something that can be classified into whether they are pathogenic or likely pathogenic, or otherwise in the other end of the spectrum, just benign variants of those individual genes. So the list of inherited cardiac condition genes are really well laid out. We have decades, at least 3 decades of research that have already laid out all these genes individually. A panel that we frequently use now comes from Invitae, and you can see some really famous genes, MYBPC3, MYH7, the troponin genes, the lamin AC genes to do with cardiomyopathy throughout. And then, of course, the one that we are referring to today, which is TTR. So definitely have to talk about amyloidosis. Yok-Chin described how it's an extracellular deposition of abnormal protein fibrils. As she said, there are at least 20 to 30 different human proteins that are amyloidogenic. And actually, these could also be the genes that you can sequence if you suspect that they are the ones that are contributing to amyloidosis. Again, as she also elaborated, there are versions of amyloidosis, which is AL, AA, and either hereditary or acquired. In genetic testing, there are various companies that offer the panel of genes. And as I say, each one of these genes may cause fibril formation, amyloidogenic potential. But the ones that we frequently hone down on, most frequently occurring in this realm of rare disease, is TTR, transthyretin. So if you opened up the Genome Browser, the University of California, Santa Clara has a very famous Genome Browser that us geneticists refer to. The TTR gene sits on this specific location, cannot remember which chromosome. There are 4 exons with long introns lying between them. So I'm giving 2 examples of mutations that have been reported, and you can see that there are actually a whole panel of frequently occurring mutations. The way to read this is the number 30 refers to the amino acid position. And in this case that I'm highlighting, it's cysteine to arginine. If you remember your preclinical days, C, cysteine, R, arginine. It's a cysteine to arginine change at the 30th amino acid position. So in this research realm, it's been found that this specific mutation is frequently showing neurological features rather than cardiac features. And another database that we frequently refer to in genetics is called ClinVar. Again, it's something that you can access fairly easily. And you can look up this specific mutation, which is described in several different groups around the world. Families have been found. And from these labs that you can see here, Invitae being one of them, they have come to classify this specific variant as likely pathogenic, except in one case, and I think they will correct themselves now to be more aligned and harmonized. So another example is isoleucine to leucine change. So that's in 88 position. Again, example of this ClinVar database and the different labs that have annotated this particular variant to be pathogenic or likely pathogenic. Okay, so cardiomyopathy is a great place to practice genetics today. If your patient has hypertrophy, if it's not obviously down to these conditions, you can consider the sarcomeric gene mutations. If not, as Yok-Chin said, there are clear features that points to why you would be testing the TTR gene and suspecting amyloidosis. Otherwise, there are also phenocopy genetic basis, which is even rarer, Fabry disease and Danon disease. And it seems like this is really worth testing today, mostly because there is treatment that has been coming. I think this might be discussed later by subsequent speakers, but there is a move towards editing the TTR gene itself, because all you really need to edit is the gene in the liver, since the liver is responsible for making transthyretin. And there are also versions of therapy that is targeting the non-formation of the fibrils or breaking up the fibrils, as you can see here. And I think Yok-Chin has shown a slide that relates somewhat to this, in which it's showing us where genetic testing sits, somewhere down at the bottom when you would like to be able to tell if this person has a hereditary form or not, and then give family counseling. I also thought I would put a plug-in for this recent research that we did, which is testing the genetics around hypertrophic cardiomyopathy patients in Singapore. It turns out that there are 4 genes that predominate the basis of this hereditary condition in hypertrophic cardiomyopathy: MYBPC3, MYH7, and 2 troponin genes. We haven't found any new genes yet, and it was also interesting that we didn't see any mutations in the TTR gene for this particular group of people that were recruited to this study. In the general Singapore population, there was the SG10K whole genome sequence effort done on healthy individuals. You can see the healthy individuals are broken up into the ethnic groups. Again, we took these whole genomes and looked for mutations that sit in genes that cause inherited cardiomyopathic conditions. In this column you see here. And again, it was interesting that in this case, the condition that stuck up the most was familial hypercholesterolemia, where we found that there are at least this number of people, 1 in 281, 1 in 117, that have mutations in genes that would be causing them to have suspicion for familial hypercholesterolemia. And in the context of TTR, we found that there were 5 of these variants in our healthy Singapore population with variants in these positions of the TTR gene. And in this instance, there isn't sufficient evidence to call these pathogenic. So they are coming down with the categorization of variant with uncertain significance. So then finally, the plug for 2 big policies that have been put out from the Ministry of Health. For anyone who is practicing genetics or wanting to practice genetics, I'm sure this is something that you're aware of, how they are guiding us on the levels of genetic testing, who can order tests, how research test results is restricted in use, and how we would be dealing with predictive testing. So Yok-Chin put out a series of red flags for how you would think about, come to think about cardiac amyloidosis in the patients you see. My red flags are for who you would consider genetic testing for if your patient has a relatively early age of diagnosis, if there are multiple family members with very similar condition, or especially even if there's a history of sudden cardiac death or unexplained death in the family. So I've come to the end of my slides. Just wanted to put a plug here to thank collaborators, especially you and Ashley, who's a very big geneticist, cardiac geneticist in US, which has given us a lot of training experience in this space. Thanks very much.